if you're here for the Enlightened Soil webinar, you've come to the right place. If not, maybe stay because you might learn something exciting tonight. And we'll start with some introductions and then we'll move into the webinar. So my name is Camille Newsom. I'm the Director of Grower Support for Enlightened Soil Corp. I'm also a livestock farmer up in Grand Rapids, Michigan. And I am super excited about this community that we're forming online and in person, as some of you all have met with us at growers meetings or conferences or on the farm. So thank you all for being here, and we're super excited. Please write down questions you have, throw them in the chat. This is meant to be a discussion and conversation with you all. So we encourage you to do that. Yazzie.
Hi, everyone. Thanks for being here, and thanks, Yadi, for joining us. I'm Yazzie, and I am the marketing gal for Enlightened Soil. So all of the flyers you've seen for this, it's me.
Great. And we will introduce Yadi Wang in a little bit. So we're going to start with just a little bit of an overview about EnSoil for our new folks that are on the webinar tonight. And if you've been around for a while, it's always a good reminder just to think about what the algae is doing, this amazing organism that we're working with. So we'll start with that. And then we will hear Yadi's presentation. And then we will have, as I said, a Q&A and a discussion amongst all of you. And finally, we always encourage you to share what you've learned in this webinar with your local communities, because that is our goal, is to spread knowledge and spread whatever we're learning here and the questions we're asking. So with that, we will start with just a little overview about how EnSoil Algae works. So the strain of algae that we are working with here at EnSoil is called Chlorella vulgaris. That's the specific species. And this specific species works as a powerful biostimulant, and it encourages natural processes of nutrient cycling and soil microbial activity to stimulate and accelerate plant growth. The EnSoil algae can be used on any plants that are in soil, and it really works in these 4 ways. The first is that it stimulates and grows the microbial population and the diversity. So we're not only seeing an increase in the number of soil microbes, but we're also seeing an increase in the different species, rare species included in that. The second way is that it increases plant nutrient uptake, and it stimulates overall plant growth. There's a lot of nutrient cycling that goes on with the EnSoil algae, and specifically the relationship that it has with the soil microbes. And then we're seeing that value, that nutritional value of crops grow as well with the use of the algae. Third, the algae enhances chlorophyll. Chlorophyll is that green pigment that you see when you look at a plant that's green. So it enhances that, and it also improves the photosynthetic capacity. So one of the really awesome things about the algae is that it actually gives its chlorophyll to the plant. And that's one of the reasons why we see this enhancement in chlorophyll specifically. And then finally, EnSoil algae increases plant tolerance to stress. This can be biotic stress or abiotic stress. So we see things like drought, salinity, and I know that Yadi will talk a little bit about drought specifically tonight. And then we also have seen some research on the biotic stress, the immunity response to different pathogens in the soil, specific bacteria and fungi. And then we also see an increase in the soil water retention capacity, so that soil is able to hold a little bit more moisture in it, which again, we will learn a little bit more tonight talking about Arizona and the climate down there. So these are the 4 main mechanisms, and I'm more than happy to talk more in depth about this. If anyone has specific questions, please bring them up in the question and answer section tonight, and we would be more than happy to talk about those. So we are going to move on to Yadi's presentation. We have been working with Yadi now, doing some research in Arizona at Oatman Flats Ranch, and he has been a really amazing part of our research team, and we are super excited to have you here tonight. So thank you for being here with us, and I will turn it over to you. We can't hear you, I don't think.
Is that
Yep, that's better. Oh, we can't hear you again.
Let's see.
That's good.
How about now?
Yep, much better.
Okay, yeah, technical difficulty with my earbuds. Um, I was hoping to sit in my home office and then, have some leisure time, but just like any other farmer, every day is a new day. So I literally just got notice to start this farmers market 2 days ago, and then I just finished the farmers market today, so I have a little bit wet sunshine on my face. Apologize for that. Yeah, every time when people tell me like Dr. Yadi Wang, I kind of feel guilty because I really don't fix people's health. I work with physicians a lot. I really just trying to figure out how to make soil work, and really a doctor, probably a doctor of soil. That's probably more appropriate. And Camilla, just quick question: can you help me drive the pages?
Yes, I
Okay.
will drive the pages, and if I'm going too slow or too fast, just call me out
No,
on it.
no worries. Well, appreciate that. Yeah, so I really just want to give a brief overview on what we have done so far down at the Southwest. I think is everybody think it's a challenging environment, which I quite frankly had a lot of joy in the past 3 years on this journey. Bring a lot of life back to the land, to the ecosystem. In the past 3 years, we had seen over 150 species reappeared on this land from a devastated, fallow land, 12 years that nothing would grow. That's why my title is Be Live or Not. So it's our choice that how we can work with other species in this ecosystem. It's a food supply that is how we can produce for ourselves, but equally is how we can provide for other species, allow them to help us to do the work that we otherwise wouldn't be able to do. Please. Next, please. Yeah, so the food supply and demand in the ecosystem, this is what on December 5th, that observation I've had, and I had a group of food system students came to visit farm. And then anytime I have opportunity like that, I always kind of trying to put people into labor force because I personally don't have enough labor. And so I had 30 some people. I just sent them to these 76 acres of the field and asked them to count ladybug on each plant, and then come back and then share the knowledge how, which plant. So what is the minimum number of the ladybug on each plant that they observed. And then so the result was really stunning. 76 acre on a spatial distributed field, nobody count has less than 4 ladybug on each plant, and that's in December 5th. And on the right is the picture that we all observe is a beehive that really formed on the top of the sorghum stalk, and that plant was eaten by sheep through the grazing process 2 days after, and then 3 days later, I found identical type of beehive in another part of this farm, this part of the acreage. So that tells us the nature is really resilient. Once we provide everything that is needed for the food supply and demand for the ecosystem, they can turn over themselves and then find that abundance and then going through that positive feedback loop. Next, please. But when we are looking at our human society, the food supply and demand we created, and for the farmers here in the room or people who are in the food industry, and we all know farmers really don't get much dollars. And this is 2022's data, and then farmers only got paid for 8 cents of this whole food dollar, every dollar consumers spend in the grocery store or restaurant, and that's 8%, less than 8% was going back to farmers. And stretching out this spectrum. And next, please. And if we really think about, if we want the biggest topic I think we need to reconsider is we always have this mentality with the growing population want to feed the world. And living in a border community, Nogales is the largest land port in the United States. Every single year, 9 billion tons of the produce coming through that border, going through up north New York State as the furthest state is going through 3,300 miles travel. And 6 million tons of those 9 billion tons of the produce, yes, statistically less than 1%, but that going to waste, that is, and literally sitting there generate methane, and that's a much more aggressive greenhouse gas compared to carbon dioxide. And from that carbon footprint perspective, and that is very unacceptable. How can we reduce the waste? How can we reduce the carbon miles? And is the national supply chain working for us? And that is some kind of rhetorical question I always ask. And then, do we really need an approach to make more food, or we supposed to do something better that is produce and products coming in are harvest ripe and that is more nutritious value for the local community, and we cut that the whole supply chain mileage and carbon footprint at the same time give people confidence on where their food come from. Next, please. Phoenix is one of the largest growing city, and in this case, it has nearly 6 million people right now. And if we count all the winter birds, and a lot of my farming friends that from Midwest area come here, have their winter home here. And I said, wow, you guys really have the luxury, because we in Arizona don't have vacation time because we grow things all year long, and we don't have a winter break. But if you're looking at this spectrum and the diabetes rate and the food security issue, and all these are stunning numbers. We're talking about a city. It's not a food desert that has so high rate of obesity and diabetes and food insecurity for kids. And when we think about all the food desert across the United States, what numbers are we looking at? Is there a reason should be that way? I don't think so, unless, but the system is setting up like that, our food system. So that's something we really want to work on and trying to figure out how can we help on that issue. And next, please. The way we follow the farming method, we go through one sole goal is trying to figure out how do we restore the carbon cycle and water cycle. Essentially is the three element, carbon, hydrogen, and oxygen. Those three life building block that essentially that can help rebuild the ecosystem function, and also the plant health is part of the metabolism for human, for plant, for animal, for all living species. And if you're looking at plant, animal, and human, all those three different species that together, we have so much similarity. And 93% of the main source of elements is those three elements. Next, please. And so for us, and we always want to emphasize the 6 principles, context is so important. Next, please. And within that context, and then we can really hone in onto all those 5 different other principles. How can we maximize on using the practices? Practices will always change, and really hone in to maximize all those principles as much as we can. Next, please. For the farm, our context is we are at 500 feet from the sea level elevation, 4 inches rainfall annually, hottest day 125 degree. We have a lot of wind pressure, so basically all other farmers have been talking about. But for us, the invasive weed pressure is sometimes create a lot of opportunity for us through the ecological succession process. 2018, the owner purchased the land from family's hand. It's a rundown farm, almost sleep out of their 4th generation, and nothing would grow on there. The soil is sterile. It's been followed for 12 years. And to date, the right picture is 2023 that we have recreated life, and we use very minimal amount of water irrigation for a certain part where we had a conservation cover. We have the agroforestry established while using almost half of the water for our main cash crop growth compared to neighbor farms. Next, please. And when I started this regenerative agriculture journey, all the talks I've been to and most of the people told me, and the lessons I've learned is no-till cover crop, no-till cover crop. So that's kind of the first thing I've tried is cover crop, multi-species cover crop. And to date, we have warm season cover crop, 14 species, cool season cover crop, up to 18 species. And this winter, I just put in my first experimental test at 33 warm season plus cool season species plant in at the cool part of the month to only allow the cool season crop coming out. And by the weather is warming up, and then we can have the warm season crop relay out. And this method really to try to increase the forage capacity for our sheep grazing while they're trampling through the land to help us to really add more nutritional benefits for the soil. And one thing I want to mention is these single practices is only a one practice, but look at how many different principles it can cover. And for one unique perspective on our cover crop, what we do, we don't terminate at when they're flowering and then trying to kill them. We terminate them at when right before I plant my cash crop in. The reason is because I want to maintain that living roots, the microbial integrity. So when I have water, plant seed going through in this field, and the microbe can start working. So maximize living roots is always my primary goal, to turn over the land, turn over the nutrient capacity. Next, please. And speaking of sheep, and then in this picture, I want everyone to notice that one of the most invasive species we have 14 species here in this cover crop species in here, but predominantly this is a poor part of the worst part of the land. We have a lot of Bermuda pressure. And even though Bermuda provides so much more nutritional value for sheep and grazing capacity for sheep to allow them to benefit from that. And Bermuda grass is very bacteria-driven plant. Throughout the ecological succession, I've seen there are success cases in our field when the fungal community improve, Bermuda start facing away, and then all the other species start coming back in. Again, by having the sheep, they are an excellent pruner. They essentially, we only allow the sheep no more than 3 days of the grazing period. We try to move them on a daily rotation. And the goal is to prune the plants to certain size that so they can regrow, and to allow that, maximize that root capacity to expand, to host more microbes on the biological cycle and nutrient cycling capacity. Next, please. And sheep, we also use sheep as one of our termination method, and the other one is mechanical termination method. Roller crimper has been developed in Wisconsin first, and then to specifically terminating cereal rye. And for us, when we had roller crimper, we had no idea if our crop would work or not. So that's why we've been testing all these different species and trying to figure out the crop height and the integrity of the plants and how thick of the stalk they should be. And so far, if we have anything that boot stage high, if we can maintain over 50% of the tall sorghum, Sudan, those type of tall grasses, and everything else broadleaf spread out in the field, we can actually crimp down all those tall grasses to choke down everything else. Have some success cases on how we can, use the roller crimping termination to kind of eliminate some of the those cover crops that we don't want to create competition or resprout really aggressively. Again, we're really trying to crimp right before, maybe 1 week or 2 weeks before we are planting our winter wheat into the ground. And because we really trying to hone in, trying to maintain the living roots as long as we can, because we're really brutal environment. Once the microbe going dormant, to wake them up is not as easy as other places where it has received a lot of rainfall. Next, please. Heirloom seed, we grow heirloom wheat, and the genetic expression of this type of wheat is outstanding. And wheat in general, the modern wheat has up to 44 pairs of chromosome, and the genetic, the genes that pair into those chromosomes have only one single goal, is for the quantity, for the yield. Whereas the heirloom seed, they only have 14 to 18 pairs of chromosome. Not being a plant scientist, I always say less is more in this case. And then so we really trying to allow them to express genetically, to adapt into the environment, and to really produce something that has a unique profile of the protein and gluten that which is weaker in this case, is helping people to digest better. Now, the epigenetic is so important. And our seed saving program, I learned that from old grandma in the indigenous community, and they have thousands of years of practice and saving seed for different years of seed, combine them together to plant into the field. And I'm doing exactly the same thing. I don't plant, let's say, last year's seed alone into my field, but instead I plant at least 3 years of this mixed seed of the same species. Let's say Red 5, which was orange, New World landrace variety, first grown in Wisconsin, developed in Canada, and then it worked really well here in the Southwest. And if I plant that Red 5 species in the field, I always plant 3 years, of the wheat, Red 5 seed, blend them together and plant in the field to really allow that epigenetic and the genetic expression hone into the environment, and to have that information exchange over time. And so really, and also from the root structure and the information sharing on the cellular level, and that will create a different abundance for the microbial content and contacts. And that from the maintaining the living roots diversity part on the biological cycle, that is something is very unique, which is all coming from the epigenetic side. Next, please. Integrated pest management. I've showed a picture of ladybugs and bees, and that's a natural cycle. And we also had a lot of gopher issues down in the Southwest. And then on the bottom right picture, you see that big hole. We currently still using flood irrigation. And so one gopher traveling through the river channel to the field, and create a lot of those holes underground when we irrigate, and we enlarge those and then causing trouble. And I almost stuck my tractor in that hole, but I kind of was lucky to drive away. But that's kind of the problem we have. And what we've learned is barn owls can eat gopher-sized rodent, eat about 1,000 of them in a year, and they're very productive. And a pair of barn owls can give 5 babies, birth 5 babies every year. So that's really how we want to use them as integrated pest management, and so that we don't have to use methods like fumigation, which is kill all the fungal environment in the soil, And when we lose the fungal environment, that's when we have the problem to maintain the living root structure. So going back to that, one of the principle, that's something we really want to going back to see how can we maximize our capacity using alternative method to maintain the subsurface structure, physically, biologically, and so that can chemically turn over the nutrient and helping us to restore this nutrient cycling in a better way. Next, please. Next, please. Yeah, so 3 years ago when I started this journey, we Camilla, could you go back to the previous page really quick? The previous page. Yep, this page. Thank you. So next page. All right. Yep. So Desert Southwest, this is the irrigation flood irrigation schedule we're looking at. So 3 years ago, for the same field, I can irrigate from canal to the end of the border, call those border system in between two black line in 5 hours. Same amount of water as of today, I can water 3 days. It wouldn't go past 3/4 of the field. And because of water infiltration, that increased dramatically. And on the left side, you can see the soil moisture. Those are 7 days after irrigation. Right now, I can have water holding capacity at up to 5 weeks in the brutal desert environment. This is talking about April or March or April, when the ambient temperature can go up to 105 degrees. And then you see the soil root structure really helping to that fungal hyphae is really helping glue everything together to keep the integrity of the plant roots, soil, and microbes all together. And this is something we really think the living root is so incredibly important on these principles. How every time we think about how can we maximize our regeneration journey on the farm, and living roots is always come back to my mind is one of the critical part. Next, please. Yeah, and then this is the image of July this year. We got 1/8 inch of the rainfall, and 2 days later, I did this analysis. On the left side is the bare soil, and on the right, in the middle of the picture is soil with Bermuda grass. I always want to show people Bermuda is not so bad sometimes. And then, so if you look at those pictures, bare soil in 2 days, this is top inch of the soil. There's no water retention. Whereas look at the Bermuda grass, think about that, in and out, 1/8 inch of the rain that can give me 1 inch of the moisture content retain the soil in 2 days. And that's 5 a.m. I collect those samples, 87-degree ambient temperature, and look at my boots and everything. I have dews on the ground. And that small water cycle and how to build that and how to keep the water vapor and low in the low atmospheric level, and then be able to retain that, putting back down to the soil, to the plant. This is the critical part of the water cycle. And those Bermuda grass, even, they're the part of the carbon cycling, because remember that plants, human, and animal, we all have carbon, hydrogen, oxygen. That's the three main elements to build the cellular structure to establish life. So carbon cycle and water cycle, we cannot separate them to talk about one thing or the other. They have to be expressed together. Next, please. Next. Yep. And this is another example, is similar test in November this year and last year, and then ambient temperature was 70 degrees, and this is collect the sample 2 days after the 3/4 inches of rainfall, and bare soil, and in the middle is the dead vegetation, no living roots, and the right side, living roots, Bermuda grass in my cover crop field, and 14 inches core we collected. And then in the next picture, next slide, please. Is that going? Yep. So if you look at this next slide, and then on the left side, bare soil, you can see those powder, no soil structure slipping off from my hand. There's no soil moisture retention. We're talking about November, 70 degrees, and 2 days, nothing has left in the soil. And in the middle part you can see there's partially soil water retention, but it's not fully. This is no living roots. There's no microbial stimulation. There's no plant and soil interaction. On the right side, even in the Bermuda field, the soil structure and the moisture water holding capacity in 14 inches, that's the amount of moisture we have. Remember, we only have less than 1 inches of the rainfall, and the input of the water from the rainfall, there's no irrigation, pre-irrigation whatsoever, right? The only possibility for that moisture profile in the soil is the plant root exudate. When the plant on the cellular level sense there's some rainfall, they want to be able to leach some of the material to incorporate those moisture together to turn over the nutrient and recycle the nutrient to reuptake for the plant growth. So this is the only possibility that can explain this type of phenomenon. Next, please. So yeah, with that, I really want to just going back to that very beginning of that topic is the food system. How do we look at the food system through the length of water cycle and carbon cycle? Are we okay with producing everything, even in a regenerative setting, but working in a commodity system that not only have a lot of carbon footprints, but also sending, particularly like us, sending the water right away to other places, not serving our community, versus, if we're working in an alternative structure and then building some sort of pipeline and then working with the local community and bring the nutritious, valuable product back to the community, and everybody can activate their food dollar and vote with their right for the water cycle and carbon cycle. Drought is just a symptom of the broken water cycle. Erosion is a symptom of the broken carbon cycle. And the water cycle and carbon cycle, it should be part of our diet, part of our metabolism of human health as well. So this is what the journey at Oatman we're really trying to steward on and work on. And I hope everybody enjoyed this conversation and open up for any questions and feedback, and trying to learn from everybody as well. Thank you.
Thank you, Yadi. We really appreciate that. And we'd love to hear, I know we got a question from Steve Wang in the pre-webinar about your work with EnSoil specifically. Would you speak to that just a little bit about your journey with us and how that maybe integrates into your regenerative practices you've shared tonight?
Yeah, so algae it's a very, in my view, is a very miraculous species. I would call that species because it's living organism. And algae not only it's kind of from a cellular structure is a polysaccharide, and which has a one side is hydrophobic, the other side is hydrophilic, and then so the hydrophilic side is lighter, is always pointing upwards, and hydrophobic side is pointing downwards. So from the physical perspective is when you think about, we have the irrigation going into the soil, and then the hydrophilic side will attract that water molecules and then pulling the water down profile and going to the soil, and that create a moisture, that create a microbial activity. And that hydrophobic side also, on the other hand, prevent water to evaporate from the soil. And so those the breakdown of those algae are the main food source for microbes to establish. And so from all those indications to us is, these are probably one of the better natural processes, and of all the type of inputs that commercially everybody is producing. I think the biomimicry part, and how can we use the species that are living that to be able to help facilitate for this whole food supply and demand in the ecosystem. That's where I'm always intrigued about it. And also Dr. James White and published those paper and talking about, oh my goodness, these endophytes that are in the plant leaf are being transported and through these the soil roots, plant shoots to the plant leaf, and then be able to fix nitrogen within the leaf rather than within the soil. How amazing is that? So I think those are indication. I think evidence are showing this species can be a one of the component for people to use to regenerate, until hopefully at one point we don't have to rely on continuous application. If we done everything right, the nature can fix itself. Now, for our experimentation, where we have done with EnSoil, we've trialed in our summer cover crop growth. And what I can tell just based on my naked eye is they are same number of times. We only irrigate one time for our summer cover crop, regardless how harsh of the environment is, because we want to train them to be more resilient. They are literally, I think, double the size of non-treated soil where we have. So I know for the fact in that there's two parts that we're looking at from the scientific perspective. Number one is they increase the microbial activity in that sense, and that could allow the nutrient cycling and all those capacity. Number two is they hold the water in a different stages, and then creating more available water that can be in a phase, in a space that plant can access. So to combat the stress over time. So I think those are two key were from a kind of logical perspective, the most fundamental part I could speak on for my experiences.
Great, thank you for sharing that. And that's definitely consistent with what we're seeing from a lot of growers. So it's great to hear your perspective specifically for your summer cover crop. So we'd love to open it up for questions and discussion from all of you. Feel free to unmute yourselves or type in the chat, whatever you're comfortable with, and we will go from there. And feel free to ask questions about anything. Yazzie, we could start with the rest of Steve's question from the submission.
Okay, yeah. So Steve's whole question was, can you tell us, well, it started with Yadi, you have massive educational accomplishments and years of experience in the field. Can you tell us what the greatest advantage of using EnSoil is? Is it the living cell, the cost per acre, the ease of application? In your mind, what sets EnSoil apart from other biological stimulants?
Well, there are so many, I think, product out there is to increase activities, right? But I think in my perspective, when I'm looking at biodiversity, I always think about how do we define or kind of think about biodiversity? Are we only thinking about living matters on the surface that we can see? Or do we consider microbial activities and living matters in the soil that we can't see and that they're providing a purpose? And what kind of food source do they use, right? The plant eat the nutrients. Well, essentially, we can simplify it like that, use the most layman term. And microbes essentially, use the carbohydrate and to digest that and then be able to grow and their community to be able to cycle nutrients and other things, which is the waste product from the microbe, right? That become a resource for the plants. And everything I'm looking at is if we're just trying to sterilize, like adding some stimulants and then into there's no difference than human uptake any of these supplement vitamins. And if our metabolic is not established for that, I like to use vitamin B12 as a perfect example. The most expensive vitamin in the store we can buy. And what I've learned from Dr. Naisha Winters, who is the integrated medicine oncologist, she taught me human body have 5 processes has to be sequentially happen in order for us to take the vitamin B12 and absorb that into our system. Otherwise, it will flush through from our urine. That's waste a dollar. And think about that metabolic part is those are stimulants. Those are vitamins that are non-living. And it is what the living cell and living matter is the most important. And to encourage each other to have this symbiosis relationship. Now, in the soil, when we're thinking about soil part, where I see the most important part is what other living organisms we can add in there and to be able to create this holistic cycle. And the EnSoil, as far as I know, these are living algae we're adding in there. These are not some of the stimulants. These are not some of the synthetic stuff that we just change a term and to make it and then try to make money. And there's some biological companies out there have trying to ask me to use product, and the first question I asked, what type of product ingredients you have in there? Was they said, well, this is proprietary, we can't tell you. I say, well, if it's proprietary, then what you tell me means this is the formulated process. And that means it's the fixed, not a complex. This is something that you know exactly what the ingredients in there. And if that's just a supplement, then why do I need that? It's not what the nature needs, it's what we want to get. But what we have to really try to do is to learn what nature wants and what the nature asks us to facilitate, not to dictate. And that's my job as a land steward. Our yield sucks. I always tell people that. Don't ask me the yield question, but that's not my focus. My focus is to build a healthy system for every living organism surround me, and also to help my own organism, the human health. And that's the part of the journey on this work that we're working on.
We thank you for doing that, Yadi. That's such an important, just that's the crux of what we're all here to do. So I really appreciate you saying that.
I have a question. Hi, everyone. Hi, Yadi. Thanks for being with us tonight. I was wondering if you were planning on adding any other cash crops to the farm this year, anything interesting that you've seen in the seed saving or in the community that you might be adding for your next season.
Say that again, that question.
I was wondering if you were thinking about adding any other cash crops or anything interesting that you've seen in the seed saving work that you all are doing. Or...
I'm glad you asked that question because we grow heirloom wheat. It sounds pretty specialized, but really, to me, it's like wheat is what we started that to help to save a little bit water and to hopefully generate a little bit of revenue, but we're still trying to learn that because the marketplace is so messed up right now. But it's a low-archy crop. It's C3 plants. Now everybody talk about photosynthesis, talk about how the carbon drawdown, but nobody really talk about photorespiration. C3 plants has a very high photorespiration that lose that respire the carbon dioxide from the plant system and equally lose the water vapors. And VPD in the research world, people say that vapor pressure deficit. And that's a huge deal for these C3 plants that what can provide it. For me, the new journey right now, I'm really trying to focus on how can we grow some C4 plants, some canned plants, C4 plants as milo, millet, or even hemp, right, for fiber. So those are the opportunity. I'm talking with our Department of Agriculture here, trying to figure out how can we grow some hemp for fiber. I'm working with natural building builders who use hemp for hempcrete fiber and hemp walls for insulation and hemp rebar, which create a lot. Hemp by itself creating a lot of biomass, turn over the carbon. It's a C4 plants. There is very limited photorespiration, almost no photorespiration from that whole process, and the metabolism process, and that helped the carbon drawdown. At the same time, is a lot. Don't create a lot of opportunity to lose the water vapor in the brittle environment, particularly like ours. And also it's very resilient. A little bit weak pressure, it doesn't matter. And if farmers doesn't have to spray, and think about, we are living in an era that all our buildings, drywalls and insulations particularly is so toxic. And we're living in a toxic box. No wonder we get sick. If we're building with this hempcrete material and all those building material, creating a breathing system, that we're living in a breathing system, that's an alternative way to help people get into a health. And because farmers, we do food, fiber, and fuel. That's three things we grow, right? And we suck. We just have to rely on all those things. Nobody will be better to go walk on the street naked, right? And everybody have to drive. We all have to eat. So how can we do better on creating all those three materials, and that could put us in a different position? That's kind of my next journey, is like, how can we grow more C4 plants and then to really retain more water and into the low atmospheric level, and then to retain and build that small water cycle on the ground in our brittle environment.
Yeah, thank you. More atmospheric rivers.
That's the goal.
We have another question in the chat from Paula. Is EnSoil Algae something that a home gardener can have access to? Definitely. You can use EnSoil Algae in your home, on your houseplants, in your yard, on your lawn, raised beds, any type of home garden. Definitely, we have lots of folks, myself included. I apply it on my plants every time I give them water. So that's for sure. We sell the algae in different size bottles, appropriate for home use as well as large scale agriculture. So we're happy to answer more questions about small scale use if that's something that you're interested in, but that's a great question. I'll ask you another question, Yadi. As you were talking about the water infiltration and that graphic that you had that had the two sort of vertical tubes going down into the soil channels, I guess. I'm curious how you see you talked a little bit about the hydrophilic and hydrophobic parts of the algae. I'm wondering how you've seen any changes in that water infiltration or the moisture retention from using the algae on your summer cover crop?
I didn't test that. I wish I would have done that. I in theory, I think there is a slight improvement because from how the water uptake and water redistribution for the plant growth, that's an indication of that. But in general, like if we're creating a lot of biomass, a lot of living matter in the soil and holding that soil integrity, this minimized disturbance is not strictly talking about no-till only. It is about minimize the physical disturbance, which is tillage, and chemical disturbance, less chemical input, and biological integrity. Right. So is whereas the living roots and the microbial, community, all those things. So I think all those things play a part of these. It's so complex, it's so heterogeneous, and it's not one thing. It's not a silver lining that we can save the whole world. It has to have everything happen sequentially or together, just like the vitamin B12 example that I've learned from the physician I'm working with. So I'd say it's complementary. We shouldn't think about that as a gold standard, but it is something, if that has the purpose that we can help improve and at the very beginning of the stage for people to transition, and that's what we should be hopeful for.
Thank you. It's great to see the integration that you have with all of these different regenerative practices. We always like to remind people, and you're such a living example of this, is obviously, and as a farmer, there's not one thing that's going to solve all of your problems, or fix your land. You gotta put your eggs in a lot of different baskets, and it's great to see that you have all of these different ways of working with the land and stewarding the land that are using all of these regenerative models. So I'm glad that, thank you for talking about that.
Yeah.
And I'm glad folks can hear that.
Appreciate that. I have a question actually, probably from a producer's standpoint, because I'm going through a irrigation conversion, from the flood irrigation, because I really messed up that system, no longer work. I can't even water my whole field, evenly distribute the water. I create anaerobic and aerobic condition in my field. And now my new conversion will be sprinkler irrigation. And one thing I didn't consider about a microbial, like the living cell integrity, is the pressure of the water coming out, or pressure regulator, how those coming out will mess up with all these biological living matter, right? Because I plan to fertigate through the sprinkler system to apply this algae going spray out, just like a foliar spray, right? One question I have is, have you guys kind of discovered or do some and did any study about the algae cell integrity, and then go against the pressure system? What kind of pressure will actually be the threshold for them to become dead or living, in that sense?
Yeah, that's a good question, and I can speak to some of that. And then Mary, if you want to talk a little bit about the research side. But I know that, for example, when I go out and spray the algae, I'm using a tractor pulled sprayer that is pressurized, by the PTO on the tractor, and so I'm increasing that pressure up pretty high to get the liquid to go through the nozzles and the output of the sprayer. And we've had no problem with the integrity of what the algae is doing and the benefits of it. In terms of research studies on that, Mary, do you want to speak to that a little bit?
Whoops. Oh, hey, can you hear me?
Yeah.
Hi.
Yeah.
Yeah, we have, I'm sorry, I missed the first part. I was having a question asked of me at home. But as far as research, are we studying the pressure that the algae can withstand?
That the algae can withstand? Is there any integrity issues with high pressure?
We haven't had any integrity issues in the systems we've been used in. The algae being a pretty tough cellular structure is why it's used in agriculture, is because it's a pretty It's been proven to be a pretty hardy, able to withstand. And then, you know, as our phycologist says, is basically the cell body is like a time release because it can withstand. We do have a couple of our partners in the region and growers in the region that are looking and using different nozzles for spraying biologicals so that they can spray the algae with, you know, with other compost teas and vermicompost teas, and lots of the biology won't be damaged there. So we've talked about actually having one of these webinars focusing, I know Randy's on this call, around the equipment that people are using in order to incorporate biologicals into their systems. I think we should follow up with that in one of these webinars. I think that'd be interesting to do.
Yeah, well, let me know, because I'll be curious on the highest pressure threshold that will actually, you know, the living cells can withhold and withstand, because it's so important that the living matters that we put into is not just a dead cell as a part of the food process. And because if we think about how we messed up is because we eat a lot of processed food that is not no living cell, right? And the same thing is for microbes and for the whole environment. If we only provide them the dead processed food rather than giving them the living cell, and that's the significant differences.
Mm-hmm.
Yeah, that's a great question. We have about 2 minutes left, and we have a great question from Randy in the chat. So Randy asked, how has Yadi applied algae, and when does he maybe do this on his regular crops?
So far I've been just following the instruction. Believe it's 8 ounces of the algae per acre. That's how we apply, and then we use flood irrigation, did this trial. So basically I fertigated into the field and let that dilute in the natural way through the canal irrigation to see that effect. And I'm still trying to developing method, and then I'd love to, you know, kind of continue using this product. And I think the great question I need to understand is the cellular integrity on the pressure threshold, because my current development of my irrigation infrastructure conversion, optimum, you know, pressure is a solid set sprinkler system, mobile solid set sprinkler system for the flexibility. So the lowest pressure I can run is around 35 or 38 pounds, which is much higher than the pivot or linear set of sprinkler set up at 15 to 20 pounds. So I really want to understand that, so that you know, before I apply, because to me, even algae, they're living matters. I want to respect that life, and you know, putting in the right context. And so I want to find ways to modify in a degree to apply in the right format.
We will definitely explore that for you, and get you a little bit more information on that as we learn about it alongside you. But thank you, Randy, for the question, and thank you, Yadi, so much for your time and everything you've shared with us tonight. We all really appreciate you being here. So all of our gratitude goes out to you and everyone that's joined. If anyone wants to stay on and ask any more questions that they have, Yazzie and I will be here for a couple minutes. We're also always happy for you to call us, and we'll talk to you anytime. So with that, everybody have a lovely Thursday night, and yeah, big thanks for being here.
Thank you for the invite, and I look forward to work with you guys and with the community here in the future. Feel free share my email address with anybody who has specific question, want to follow up, and I'm always happy to learn from each other anytime. It's a lifelong journey. Otherwise, it will be a living fossil rather than living life.
Definitely. Well, thank you so much, Yadi. We will share your information, and have a wonderful evening. Rest up after your long day of farming. I know what that's like.
Thank you very much.